Hi everyone, am I audible? Hi everyone, I'm a Nextillo faculty for dermatology and I'll be teaching you the basics and concepts of dermatology which will be helping you in getting most of the questions asked in your exams, right? So, and I thank Mr. Sunil sir for giving us this opportunity for us to teach you and for all of you to learn from us in cracking your exams. So it's important to understand why dermatology is important for exams. So in MBBS, we have limited exposure to dermatology. And while preparing for exams, it is considered as a minor subject. So very less time is dedicated to study it and also to revise it. The students rush to complete the subject in a minimal span of time, and also while during revision they try to complete in the minimal span of time. So that is why it's critical to understand that dermatology, although it's a minor subject, but it's given more weightage in the recent exams. This because the pattern has been changed and most of the questions has been asked in concepts as well as through pictures. And dermatology is a subject where it will be very easy to ask questions because it's mostly picture oriented or like clinical oriented subject. So the question will be having the stem from dermatology, but the question will have answers asked from microbiology or from pharmacology. So it is important that to understand that dermatology, there will be at least 5 questions asked in like pure dermatology, and there'll be at least 3 or 4 questions which will be having the stem from dermatology. So there'll be at least 7 to 8 questions which will be asked in the exams. And it's also important to revise the subject, and at least 2 times revision is important because one time reading is not alone sufficient to complete the subject. So I'll be helping you in understanding the basics and concepts in dermatology, as well as I'll be through this live lecture series, I'll be helping you in the important topics which will be asked in the exams. So it is important that you concentrate on the pictures, but don't expect the same pictures will be asked in the exams. However, the morphology of the lesions would be more or less the same. So if you concentrate the pictures and if you understand the topic, you'll be able to get the questions right. So before before going to the subject properly, we should know what will you learn in dermatology and what you are expected to answer in the exams. Like what type of questions will be asked, like what is expected from an MBBS graduate. So this mind map shows the important uh, topics which is expected from you as an MBBS graduate. First is the basics of dermatology. In this, you will be reading about the structure and function of skin. And the second topic, you'll be understanding the morphology of the lesions and what are the primary and secondary lesions. And in vesicular, next we'll be talking about the important disorders. Like in dermatology, we'll be learning about these disorders like the vesicular-bullous diseases like pemphigus and subepidermal blistering disorders like pemphigoid group of disorders, and inherited bullous disorders like epidermolysis bullosa. And there'll be important topics like psoriasis, lichen planus, and pityriasis rubra, pilaris, and pityriasis rosea, which are like very important topics which are commonly asked in exams. And this is grouped under the papular squamous disorders. And that's another group of disorders called pigmentary disorders, in which you will have disorders with hypopigmentation as well as that of hyperpigmentation. So hypopigmentation or depigmenting disorders are vitiligo and albinism, and hyperpigmentation disorders include the melasma, and other pigmentary disorders is the taste, like freckles and lenticels. And the most important topic is dermatitis, and it is and it includes atopic dermatitis, seborrheic dermatitis, and contact dermatitis, which can be allergic contact dermatitis or irritant contact dermatitis. So next, like, it includes the group of disorders of the appendages, like hair and nail disorders, disorders of the glands. In hair disorders, you'll be learning about alopecia areata and androgenetic alopecia. Regarding exams, these two topics are very important, like trichotillomania and scarring alopecia. And next is nail disorders, and then there are two good subgroups, like disorders, nail changes associated with the primary dermatosis, that is primary dermatological diseases, and the nail changes associated with the systemic diseases. Next, you'll be learning about the disorders of the sebaceous and sweat glands, in which you'll be learning about acne, rosacea, hidradenitis suppurativa, and less important topics are chromidrosis and bromidrosis. The most important topic in exams is cutaneous adverse drug reactions, which include SJS, TEN, fixed drug eruptions, erythema multiforme, which is an hypersensitivity reaction, which can also be caused by other things other than drugs. And the most important topic for the exams, in which 50% of the questions from dermatology will be asked, is infections. And infections can be caused by bacterial, but it includes Mycobacterium, tuberculosis, and Mycobacterium leprae, causing leprosy. Viral infections, fungal infections, protozoal and parasitic infections, and sexually transmitted infections. You should give more importance to leprosy and STI. Okay, and it is also important to understand the basics of cutaneous tuberculosis. What are the classification? What are the disorders? And how do you investigate and treat it? And fungal infections include superficial fungal infections and deep fungal infections. And the protozoal infections include scabies, which is more commonly seen in dermatology OPD. And in exam wise, it is important to know about pediculosis, leishmaniasis, and Peter's dermatitis. So this is all about the things you'll be learning in dermatology. So today we'll be discussing about the basics of dermatology, in which we'll be discussing about the anatomy and structure of skin. So let's go to the topic for today. That is the basics of dermatology. So in this topic, today we'll be discussing about the structure of skin. So we'll be discussing about the skin layers, and what are the appendageal structures, what are they, how are they, where are they situated, and what are their functions, and the important questions which can be asked in the exam. So first is the structure of skin. So structure of skin, it's important to understand that this is the dermo-epidermal junction or the basement membrane junction. So this junction is called dermo-epidermal junction. And this junction separates the epidermis from the dermis. So the layer above this junction is called the epidermis. This part of skin above dermoepidermal junction is called the epidermis. And the part of the skin that is found below the epidermal junction is called the dermis. And in dermis, you can see there are appendageal structures, that is hair, sebaceous gland, eccrine gland, and the apocrine gland. So these are the appendageal structures that are situated in the dermis, which includes hair follicle, apocrine glands, this is sebaceous glands, and this structure is called the eccrine gland. And the part of the skin that is found below the dermis is called the subcutis. So subcutis is the bottommost layer of the skin. So there are three layers: epidermis, dermis, and the subcutis. And in dermis, there are the appendageal structures like the hair follicle and the other glands. And the most important cell in the epidermis is the keratinocyte. And the most important cell in the dermis is the fibroblasts. Fibroblasts secrete collagen and other ground substances. And collagen is the important structure found in the dermis. Okay. So we saw the dermo-epidermal junction, and above the dermo-epidermal junction is the epidermis, and below that is the dermis. And in the dermis, there are the appendage structures, including the hair follicle in the glands. And below the dermis, we see subcutis. Most important cell in the epidermis is the keratinocyte. The most important cell in the dermis is the fibroblast, which secrete the collagen. The collagen is the most important substance found in the dermis. Below the dermis, we find the subcutis. And in subcutis, as you can see, is made up of adipose tissue. So this is made up of adipose tissue. Okay. This is about the layers of skin. Okay. So this is a schematic representation of how a skin would look like. But in actual scenario, this is how the skin would look like when stained with eosin and hematoxylin stain. Okay, so this layer is the dermo-epidermal junction, and above which is the epidermis, below which is the dermis. Okay, and as you can see that dermo-epidermal junction is thrown into folds. Okay, so the part of the epidermis which is projecting downwards is called the retiridges. So this part of epidermis which is projecting downwards is called the retiridges. And this part of the dermis which is projecting into the epidermis is called the dermal papilla. Okay. So the part of the epidermis which is projecting downwards into the dermis is called the retiridges. The part of the dermis which is projecting into the epidermis is called the dermal papilla. Okay. And this is how the skin would look like when stained with eosin and hematoxylin stain. And these cells are made up of the keratinocytes, and here you can see it is made up of the collagen. So next, we'll be discussing about the structure of epidermis. Okay. So we know that the most important cell in the epidermis is the keratinocyte. Most important cell is the keratinocyte. And more than 95% of this cells in the epidermis is made up of keratinocytes. So all these cells, or most of the cells which you see here, are the keratinocytes, but arranged in the order of maturation. So this cell is different from this cell, and this cell is different from this cell, and this is different from this. But all these are keratinocytes, and these are the different stages of maturation. Okay, and skin is a stratified structure. Stratified structure. Stratified means layered. Okay, so it is arranged in different layers. The bottommost layer. So this is the Dermapodal Junction, and this single layer of cells is called the stratum basale. This single layer of cells is called the stratum basale. The basal layer of cells, or the bottommost layer of the epidermis. Okay, basal means bottom, and it's in basal layer. And it's a single layer. And it's the most mitotically active layer. Okay, mitotically active. That is, this is the layer where the keratinocytes divide and proliferate. Okay, so it's a single layer, and it's mitotically active. And keratinocytes are called keratinocytes because they contain keratin. And the keratin differs in the keratinocytes of the different layers. The base cells in the stratum basale contain keratin 5 and keratin 14. Okay, and these cells are cuboidal or columnar. So these cells are attached to each other by desmosomes, and these cells are attached to the dermis or the basement membrane through the hemidesmosome. So there are two important structures called desmosomes and hemidesmosomes. The desmosomes helps in othering the cells adjacent to each other, and hemidesmosomes helps in othering the keratinocytes to the basement membrane or the epidermal junction. And these desmosomes are made up of the proteins. The most important protein is desmoglin. And the cells in the basal layer contain more of desmoglin 3. Okay. So it's a single layer, it's mitotically active layer, and it contains the keratinocytes contain K5 and K14 keratins, and it's made up of cuboidal or columnar keratinocytes. Adjacent cells are added to each other by desmosomes, and the keratinocytes are added to the dermis through the hemidesmosomes, and desmosomes contain desmoglin protein, and desmoglin 3 is more expressed in the keratinocytes of the basal layer. Okay. Next, when these cells divide and proliferate, the cells are pushed up into the next layer, and this layer extends from here to here. Okay, so This layer is called the stratum spinosum. So why it's called spinosum? Because you see spines. The spines are the desmosomes. Okay, these contain spines. These spines are nothing but the desmosomes. And so when the cells divide and proliferate from the basal layer to the stratum spinosum layer, there is change in the expression of the proteins. Okay, so keratin 5 and 14 changes to keratin 1 and keratin 10. Okay, so in basal layer, keratin 5 and 14 are expressed, whereas in stratum spinosum layer, the keratinocytes contain K1 and K10, or keratin 1 and keratin 10. And they also contain the desmosomes. And in desmosomes, the expression of desmoglin 1 is more than the desmoglin 3. Okay, desmoglin 1 is expressed more. So this is how the keratinocyte look in the stratum spinosum. It is polyhedral shape. Okay, so polyhedral keratinocytes, and between the keratinocytes, you see the spines or the desmosome group of proteins. That is why it's called the stratum spinosum. So this desmosomes helps in othering the cells to each other. The adjacent cells of the adjacent keratinocytes are connected to each other by this desmosomes. And this desmosomes looks like spines when seen through electron microscopy or through microscopy. That is why this layer is called stratum spinosum. And it's a multi-layered and contains 3 to 8 layers. Okay. So the difference between the stratum basal layer and stratum spinosum is it's a single stratum basal is a single layer, but stratum spinosum is a multi-layer, and keratin 5 and 14 expressed in basal layer, and keratin 1 and 10 expressed in spinous layer. Desmoglin 3 is more expressed in basal layer, and desmoglin 1 is expressed more than stratum spinosum. And cells, when they move from the stratum spinosum to the next layer, they acquire granules. Okay, so you can see these cells contain the granules. Okay, so this is called the stratum granulosum. Because they contain granules. These granules are keratoiline granules. And in addition to that, they contain lamellar bodies or ordinate bodies. So what do these keratoiline granules contain? They contain filaggrin protein, loricrin, and immunocrine proteins. Yes, so they contain filaggrin, loricrin, and immunocrine. Filaggrin is a filament aggregating protein. Okay. So what happens in this layer is these proteins, along with the keratin filaments. Okay, keratinocytes contain keratin filaments. So filaggrin protein along with these proteins, they bind the keratin filaments to each other, and they form a proteinaceous envelope. Okay, this proteinaceous envelope is resistant to the external modifications in the environment, and this also helps in preventing the loss of water and nutrients from the skin. Okay, these proteins along with the keratin filaments, they bind to each other and they form a proteinaceous envelope. And this lamellar or ordinate bodies contain the lipids. So along with the lipids and this proteinaceous layer, they form the lipoprotein envelope. Okay, they form the lipoprotein envelope. So this lipoprotein envelope helps in prevention of the loss of water and nutrients. Also, they help in resisting the changes in the external environment. Okay, so this is called stratum granulosum because they contain granules. So when these cells or the keratinocytes in the stratum granulosum move from stratum granulosum to the next layer, that is stratum corneum, they lose the nucleus. Okay, it's this layer, it's an anucleated layer. That is, the cells in general do not contain the nucleus, and the metabolism is lost, and the cells become flat. Okay, so cells become flat, they enlarge in size, and they do not contain the nucleus. And this is a cornified layer. That is why it's called stratum corneum. Okay, these cells are cornified cells or corneocytes. Okay, that is why it's called stratum corneum. Okay, this is the uppermost layer of the skin. Okay, and this is the most important layer which helps in the maintaining the barrier, skin barrier. So this is all about the structure of epidermis. So the bottommost layer is the stratum basale. It's a single layer, and when the cells proliferate and divide, they move to stratum spinosum, which is made up of polyhedral shaped cells, which are bound to each other by the desmosomes of the spines, and it's multi-layered layer. And when the cells move from the stratum spinosum to the stratum granulosum, they acquire granules. These granules contain keratoidin granules and laminar bodies. The role of these granules is to bind the proteins to each other with the keratin filaments, and they form a lipoprotein envelope. And these cells, when they move to the stratum corneum, they lose their nucleus, the metabolism lost, and the cells become flat and they enlarge in size, and they are called corneocytes. And this is called the stratum corneum. So this is about the layers of the epidermis. And the thickness of the epidermis is more in the palms and soles, and it's very less in the eyelids. Like it's 0.1 mm in the eyelids, and it's around 1 mm in the palms and soles. So in palms and soles, in adjacent to these 4 layers, they also contain another layer. It's between this stratum corneum and the granulosum. It's called stratum lucidum. Stratum lucidum. Okay, seen only in the palms and soles. So this process of migration of cells from the basal layer to the topmost layer, that is stratum, from stratum basal to the stratum corneum, is called the keratinization. Keratinization is a process of migration of cells from the stratum basal to the stratum corneum. Okay, and in stratum corneum, what happens is, under the influence of the 2 proteins, that is proteases and sulfatases, these cells are cleaved and they are shed. So this is in the replenishing of the cell keratinocytes in the skin. Okay. So under the influence of these 2 proteins, these 2 enzymes, the cells are cleaved and they are shed. Okay. So this process of keratinization, that is migration of cells from the basal layer, stratum basal to stratum corneum and final shading, takes about 28 to 40 days. This is called epidermal turnover time. This duration is very shortened in psoriasis. It's just 10 days in psoriasis. So the normal keratinization process does not take place. So there will be more accumulation of the corneocytes will cause hyperkeratosis, and the nucleus is not lost. The nucleus is present in the corneocytes. It's called parakeratosis. So what is parakeratosis? It's the retaining of the nuclei in the cells of the stratum corneum. It's called parakeratosis, and it's commonly seen in psoriasis. Okay. So this is all about the layers of the epidermis and the keratinization process. I hope everyone is clear about the cell layers and the keratinization process. Next, we'll be talking about the various cells seen in the epidermis. So what are the cells which are seen in epidermis? This include keratinocyte, is the most important cell. Okay. Melanocytes, Merkel cells, and the Langerhans cells. Okay. So keratinocytes are the important cell, and they constitute about 95% of the epidermis. And these cells are derived from the ectoderm. These are called keratinocytes because they contain keratin. So keratin are the intermediate filaments. They are intermediate filaments. Okay. And this keratin is of two types. Type 1 keratin and type 2 keratin. Type 1 keratin or acidic keratin. Type 2 keratins are basic keratins. And type 1 keratins are numbered from K9 to K20. And type 2 keratins are numbered from K1 to K8. Okay. And they are heterodimers. That is, they are dimers. They exist in dimers. And type 1 keratin always binds with type 2 keratin. Okay, that is why they are called heterodimers. So we saw earlier that K5 and K14 are seen in the basal layer, the stratum basal. And that's when the cells migrate from the basal layer to the stratum spinosum, there is change in the expression of the keratin. So keratin 1 and keratin 10 are expressed in the suprabasal layer. Okay. And in psoriasis, there is hyperproliferation and there is change in the keratin, the expression of keratin, that is K6 and K16 are expressed more in psoriasis. Okay. So they are heterodimers, that is type 1 keratin always pairs with type 2 keratin. So you will see one keratin between 9 to 20 and one keratin between 1 to 8. So these are the keratins found in skin. There are also keratins which are found in hair and nails, which are different from these keratins, and which are numbered beyond K20. Okay. So K1 to K20 proteins are found within the layers of the skin. So they exist in dimers. So the type 1 keratin always binds with type 2 keratin, and you find one of this keratin between 9 to 20, and you also find one another number between 1 to 8. So K5 and K14. They expressed in the basal layer, K1 and K10 expressed in the suprabasal layer, and K6 and K16 expressed in the psoriasis. Okay. Next cell which is important in the epidermis is the melanocytes. So these are found in the basal layer. These cells are derived from neural crest. Okay. So during the process of development, melanocytes migrate from the neural crest to the epidermis. The most important role of the melanocytes is they contain melanin and they absorb and prevent the sun damage. Okay, so that is the important role of melanocytes. So they are dendritic cells. Dendritic means they are thrown into folds. Okay, so what is the function of these dendritic cells or the folds? So these folds transfer the melanin from the melanocytes to the keratinocyte. Okay, so one melanocyte transfers the melanin to 36 keratinocytes. Okay, the ratio is 1 in 36, and this is called epidermal melanin movement. And there are two types of melanosomes. One is eumelanosome, the other is pheomelanosome. Eumelanosome contain eumelanin. Pheomelanosome contain pheomelanin. Eumelanin is brown, gives brown black color to the skin, whereas pheomelanin gives red to yellow color. Okay, and eumelanosomes are spherical, whereas pheomelanosomes are elliptical. Okay. So this is about melanocytes. They are direct to neural crest. They are dendritic cells. In the process, the function of these dendrites is to transfer melanin from melanocytes to the keratinocytes. And one melanocyte transfers the melanin to 36 adjacent keratinocytes, and the ratio is 1 in 36. And this is called epidermal melanin movement. This is for melanocytes are found in the basal layer, and they contain melanosomes. There are two types of melanosomes: eumelanosome and pheomelanosome. Eumelanosome contains eumelanin, which gives brown black color. Pheomelanosome contains pheomelanin, which gives red yellow color. The other cells which are found in the epidermis are Merkel cells and Langerhans cells. Merkel cells are found in the basal layer. They are also derived from the neural crest, and they are the cutaneous receptors. That is, they sense the sensation and they sense the touch sensation. These receptors are slowly adapting. So they are found in the basal layer, derived from neural crest, and they are cutaneous receptors. They sense touch sensation and they are slowly adapting. Langerhans cells are present in the stratum spinosum. They are also dendritic cells. And the function of these dendrites in the Langerhans cells is to capture the antigens. Okay? They are immune cells or the antigen presenting cells. And these cells helps in the capture of the antigens which enter the epidermis. And after capturing, they migrate to the local regional lymph nodes and they stimulate the T cells or the T lymphocytes. And these cells are derived from the mesenchyme. Okay. Langerhans cells are dendritic cells found in stratum spinosum. Their immune cells are antigen presenting cells and they are the mesenchyme. So this is all about the cells of the epidermis. So when we were discussing about the the structure of epidermis, we were talking about a protein called desmosomes. That is, desmosomes were attaching this adjacent keratinocytes, and they look like spines. So the keratinocytes in the stratum, the suprabasal layer, which spines are called this that layer called stratum spinosum. Okay, so now we'll learn about the desmosomes. So desmosomes are the adherent proteins or the junctional proteins. Okay, so they are the adherent proteins of the junctional proteins. So they belong to the proteins of cadherins. That is calcium dependent adherents. Okay. So this is a protein complex. This is a protein complex, and you can see multiple proteins in this complex. And you can see they contain desmoglin, desmocolin, leucophilin, leucoglobin, and desmoplakin. The most important structure in this is desmoglin. Okay. So this is a keratinocyte. So this is a keratinocyte, and this is an adjacent keratinocyte. And these two keratinocytes, okay, these two keratinocytes are other to each other by this desmosome protein complex. And this is the extracellular matrix or the intercellular spaces. Okay, and they contain 5 different proteins: desmoglin, desmocolin, leucophilin, leucoglobin, and desmoplakin. And the most important protein is the desmoglin. So you can see that this protein complex adheres the 2 adjacent keratinocytes. And they also bind the intermediate filaments or the keratin filaments. So they bind the keratin filaments to the base plasma membrane of the keratinocyte also. Okay, so they contain 5 proteins. The most important protein is desmoglin, and desmoglin is of different types, and there are 4 types of desmoglins: desmoglin 1, 2, 3, and 4. Desmoglin 1 and 3 are seen in the skin and mucous membrane. Two is seen in cardiac myocyte, and 4 is seen in hair follicle. The most important proteins which we should be aware of is desmoglin 1 and 3. Okay, if the antibodies are developed against these 2 desmoglins, what happens is this protein complex is destroyed, and these keratinocytes will not be adhered to each other. Okay, and this leads to separation of the cells, and this leads to bullous disorders like pemphigus. So if they are the antibodies are developed against these 2 proteins, they lead to pemphigus. Okay, and sometimes this staphylococcal exotoxin cleaves desmoglin 1, okay, which can lead to bullous impetigo and staphylococcal scalded skin syndrome. Okay. In pemphigus, antibodies are developed against desmoglins 1 and 3, whereas in bullous impetigo and staphylococcal scalded skin syndrome, the staphylococcus releases exotoxins which cleaves the desmoglin. Okay, which cleaves the desmoglin. So this is about the structure of desmosomes. Next, we'll talk about the dermo-epidermal junction. Okay, so when we started talking about the structure of epidermis, we found an important membrane which separates the dermis from the epidermis, that is called the dermo-epidermal junction. It's otherwise called the basement membrane junction. Now we'll discuss about that junction. Okay, so now let's understand this structure. This is a keratinocyte found in the basal layer. So this is a keratinocyte, and these are keratin filaments. And in the basal layer, we found there is expression of the K5 and K14 filaments. Okay. And I also talked about the adherence of the keratinocytes to the basement membrane through hemidesmosomes. This structure is called the hemidesmosomes. Okay, and this layer is called the dermis. This layer is the epidermis. This layer is the epidermis basal layer, and this is the dermis, and the epidermis and dermis are bound together by this junction called the dermo-epidermal junction. And as you can see, there are different proteins, and they are arranged in different layers. Okay, now we'll talk about the different layers. So first we talked about the hemidesmosomes. The next layer, which extends between this layer and this layer, is called the Lamina Lucida. So when you see the skin under an electron microscope, this layer appears less dense. Okay, this layer appears very loose. That's why it's called Lamina Lucida. The layer below that Lamina Lucida, when you see under electron microscope, they are very dense. Okay, that's why it's called lamina densa. And this layer below the lamina densa is called the sublamina densa. And below the sublamina densa, you see the dermis. Okay, so there is hemidesmosome and lamina lucida, then lamina densa, then sublamina densa. Okay, these are the layers of the dermo-epidermal junction or the basement membrane junction. So now, after understanding the layers, now we'll understand the different proteins in each layer. So hemidesmosome is made up of 4 proteins, that is BPAG1. This protein is called the BPAG1. Next is plectin. This protein is called a plectin. And next is BPAG2 or collagen 17. Okay, this J-shaped structure. Okay, there are 2 J-shaped structures. Okay, that is extending from the hemidesmosome. Those are called BPAG2 or bullous fulvivar antigen 2 or collagen 17. And this red colored structure is called the integrin. That is alpha 6 beta 4 integrin. So there are 4 proteins: BPAG1, plectin, BPAG2, and integrins. There are 4 proteins for hemidesmosomes. BPAG1 and plectin are seen within this hemidesmosomes, whereas BPAG2 and integrin transfers across the Emulation. Okay. And you can see a cruciform structure in lamina densa. Okay. This cruciform structure in lamina densa is the lamin. And this blue color layer is the collagen 4. Collagen 4 and laminin 5. Okay. So this collagen 4 and laminin 5, they interact with the two protein BPAG2 and integrin, and they form the junction. Okay. So this junction occurs through the lamina lucida. So lamina lucida doesn't contain any structures per se, but they just coordinate the junction between the proteins. Okay. Since they do not contain any proteins per se, that is why they appear loose in electron microscope when you see the skin under electron microscope, and that is why it's called lamina lucida. So lamina densa contains this cruciform structure called laminin 5, and there is a background substance called collagen 4. Okay. So this is called anchoring filaments. Anchoring filaments. Anchoring filaments is made up of integrin and laminin 5. And below this lamina densa, you see this cruciform structure, or like this not the purse string structures. Okay? And the fibrillar structures. This is called collagen 7. Collagen 7. Collagen 4 is seen in lamina densa. Collagen 7 is seen in lamina sublamina densa. And this collagen 7, they hold the collagen 1 and 3 of the dermis. They hold the collagen 1 and 3. Collagen 1 and 3 are the important collagens in the dermis. Okay. So this is about the dermapodal junction. Okay. Dermis contains collagen 1 and 3 and is held by collagen 7. So there are 4 layers in the dermapodal junction. First is epidermal desmosome, which consists of 4 proteins, BPAG1, lectin, BPAG2, and integrin. Next is lamina lucida, which mediates the interaction of the proteins. And lamina densa contains 2 proteins, laminin 5 and collagen 4. And below that, we see the sublamina densa, which contains collagen 7. And this is a first string structure or the fibrillar structure, which holds the collagen 1 and 3. Okay, the collagen 7 is also called anchoring fibrils. So understanding this structure is very important to understand the pathogenesis of the bullous disorders. Okay, so try to remember the layers and try to remember the different proteins. Next, we'll talk about the dermis. The most important cell in the dermis is the fibroblasts. Okay? They also contain immune cells. Fibroblasts secrete fibers and brown substance. The most important fibers include the collagen and elastin. Now we discussed that collagen 1 and collagen 3 are most important collagen in the dermis. And among the ground substance, it contains hyaluronic acid, apparent sulfate, dermatan sulfate. And the immune cells include lymphocytes, macrophages, and mast cells. Okay. So dermis contains fibroblasts and immune cells. Fibroblasts secrete fibers and ground substance. Fibers include collagen 1 and 3 and elastin. Ground substance include hyaluronic acid, heparan sulfate, and dermatan sulfate. The immune cells in the dermis include lymphocytes, macrophages, and mast cells. So this is about the structure of the skin, that is epidermis at the dermis and the basement membrane junction. So please subscribe to Nextillo to see the live lectures, and you see much more content which will be helpful for cracking the exams. And also you can download the app and use this helping hand coupon to avail app for 3 months at just 89 rupees. Okay, so you can avail this plan for 3 months, which is available at the cheaper cost of 89 rupees. Next, we'll talk about the structure of hair. So hair is made up of this. If you see the hair, it contains 2 parts. The one which you see above the skin surface, it's called the hair shaft. Okay, this part which you see from above the skin surface is called the hair shaft, and below that you see the hair follicle. Okay. Below which you see this part of hair which you see below the skin is called the hair follicle. And hair follicle is divided into 2 segments: upper segments and lower segment. An upper segment is further divided into infundibulum and isthmus. Lower segment is further divided into suprabulbar area and bulbar area. Okay. So this part of skin, this part of hair, which is seen below the skin surface till the sebaceous gland, is the infundibulum. Okay. Next, this part of the hair follicle, which is seen between the sebaceous gland and the erector pili muscle, that is between this and this. It's called the isthmus. Okay. And this part of the hair, which is seen above the bulb, hair bulb, is called the suprabulbar area. And this part of the hair, which you see, the bottommost part of the hair follicle, is called the bulbar area. So infundibulum extends between the surface of skin to the attachment of the sebaceous gland. And isthmus extends between sebaceous gland and the attachment of the erector pili muscle. And below which you see the suprabulbar area, and the bottommost part of the hair follicle is called the bulbar area. And this is the bulbar area, is the part where you see the rapidly dividing keratinocytes, which gives rise to the hair follicle or the hair shaft. Okay. This is about the longitudinal section. If you see the cut section of the hair, suppose if you cut the hair and if you see cross-sectionally, suppose if you cut hair at this level and see cross-sectionally, it is made up of the innermost layer is called the medulla. Okay. This This is medulla. And after medulla, next layer is called the cortex. Next layer is called the cortex. And outer to this layer, you see another layer. It's called cuticle. Okay. And outer to this layer, you find the inner root sheath, and outer to that, you would find the outer root sheath. Okay. So in the outer root sheath and the inner root sheath. So if you see now the cross section, the innermost layer is the medulla. Next is the cortex. The third layer is the cuticle, and out surrounding it there are two sheets. The innermost is the inner root sheet, and the outermost outer root sheet. This is the cross section of the hair. Okay, this was the longitudinal section of hair, and this is the cross section of the hair follicle. Okay. So next we'll talk about the types of hair. There are three types of hair. So first is the Lenuga hair. So Lenuga hair is seen in the fetus and is shed in utero. They are lost in utero, and these are fine hairs. Okay. Second hair is the vellus hair, which is also fine hair found in most parts of the skin. And they do not contain medulla, they are non-medullated. Okay. And their size will be smaller than 25 microns. The third hair is the terminal hair. The terminal hair is a coarse hair. At birth, you see this hair in scalp, eyebrows, and eyelashes. Okay. After puberty, these are seen in axilla, groin, beard area, and mustache in males. Okay. This beard area and mustache in males, and these are also seen in axilla, groin, male. So these are the three types of hair: Lenuga hair, vellus hair, and terminal hair. Lenuga hair is seen in the fetus, which is shed in utero, and it's a fine hair. Next is vellus hair, which is also fine hair seen in most parts of the skin. They are less than 25 micron in size. They are non-medicated. The third hair is the terminal hair or the coarse hair, and the size is more than 60 microns. And they are seen at birth in scalp, eyebrows, and eyelashes. And after puberty, they are seen in axilla and groin. And in males, they are seen in addition in beard area and mustache. Okay, and these terminal hairs are medullated hairs. They contain medulla. This is about the types of hair. Next, we'll talk about the hair cycle. Hair cycle is of three phases. First is anagen phase. Second is catagen phase. Third is telogen phase. So A, C, T, anagen, catagen, telogen. So anagen phase is the growth phase. And the hair in the scalp, the anagen phase grows for 2 to 6 years. Okay. And the next phase is the evolution phase or the catagen phase. And the time interval between this in this phase is 2 to 6 weeks. Okay, it's a short period. And the third phase is the resting phase. And this the interval of this phase is 2 to 6 months. So it's easier to remember. 2 to 6 years in growth phase, 2 to 6 weeks in evolution phase, 2 to 6 months in resting phase. And after this resting phase, the hair is shed. Okay, that phase is called exogen. Okay, it's called exogen. It's considered to be part of the telogen phase. In this exogen phase, the hair is shed. Okay. So if you take scalp at any point of time, you would see more than 90% of the hair in anagen phase, 1% of the hair in catagen phase, and around 9 to 10% of the hair in telogen phase. Okay, so most of the hair follicle which are seen in the anagen phase, that is 90% of the seen in anagen phase, and they grow for 2 to 6 years. And the length of the hair, length of the anagen phase determines the length of the hair. The anagen phase is prolonged, then you will have a lengthier hair. Okay. The next phase is the emulation phase, which lasts for 2 to 6 weeks, and only 1% of that is found in this phase. Next phase, the telogen phase or the resting phase, which the hair spends 2 to 6 months. Last is the exogen phase, in which hair is shaved. Okay. This is all about the hair cycle. So we talked about the longitudinal section of hair, cross section of hair, types of hair, and the hair cycle. Next, we'll discuss about the glands. So in glands, as I told you earlier, you can see 3 types of glands. Okay, in the dermis. One is sebaceous gland, eccrine gland, and apocrine gland. And as you can see from the schematic representation, that eccrine glands open directly onto the skin surface. Okay, whereas sebaceous gland and the apocrine gland, they open into the hair follicle, and this in turn opens into the skin surface. Okay. And any gland will contain a secretory coil and a conducting duct. Okay, this is the secretory coil of the eccrine gland. It's a conducting duct. This is secretory coil for the apocrine gland. This is the conducting duct. Okay, so they contain 2 parts. One is the secreting part and the conducting part. The eccrine glands open directly into the skin surface, and apocrine and sebaceous gland open into the hair follicle. So depending on the type of secretions, they differ. Okay, the sebaceous gland, they secrete by holocrine, that is the whole matured sebocyte. Okay, they disintegrate and they form sebum. Okay, it's called holocrine secretion. Whereas in eccrine secretion, they form vesicles and they are excreted outside the cell. It's called merocrine. And in apocrine glands, the apex of the cell is lost. Okay, the apex of the cell is shed to secrete the secretion. It's called apocrine. So in holocrine secretion, the whole cell disintegrates and they secrete sebum. And eccrine glands, they form vesicles, and these vesicles are transported outside to form sweat. In the apocrine glands, apex of this cell is shed to form the secretion. It's called apex, so that's why it's called apocrine. Okay. The function of sebaceous gland is to secrete sebum. This sebum helps in the lubrication. And this sebum is made up of triglycerides, wax esters, squalene, cholesterol esters, and cholesterol. Okay. They contain 5 things: triglycerides, wax esters, squalene, cholesterol esters, and cholesterol. This sebum helps in lubrication. And the eccrine glands, the important function is sweat. Okay, they secrete sweat, which helps in the thermoregulation. And the sweat in the secretory coil is isotonic. Whereas sweat in the conducting duct is hypotonic. This will be taught for you in physiology. This is hypotonic because while the sweat is transversing through the conducting duct, the sodium and chloride are reabsorbed. Okay, that is why it's hypotonic. Next is the sites. The sebaceous glands are found everywhere except on palms and soles. Okay. So sebaceous glands are present everywhere, but they are not seen in palms and soles. They are seen in abundant in the face and scalp area. Okay. And sometimes these glands are modified. So sometimes the glands are modified. Like in eyes, like in eyelids, there are 2 glands, meibomian glands and Zeiss glands. Okay, these 2 are modified sebaceous glands seen in eyelids. And in breast, you can see Montgomery tubercles. Okay, this is also modified sebaceous gland. And in genitalia, you can see Tyson's glands. Okay, in male genitalia, you can see the Tyson's glands. These questions will be commonly asked in exams. Okay, meibomian gland is a modification of which gland? It's a modification of sebaceous gland. It's in the eyelid. Like sebaceous glands, eccrine glands are also present everywhere, but they are absent in vermilion border of lips. Okay? They are absent in external auditory canal. External auditory canal, they are absent. They are absent in genitalia, anogenital region. Like in females, they are absent in labia and clitoris. In males, they are absent in glans penis. Okay, these are the sites where eccrine glands are not found. Apocrine glands are present at birth, but they mature during puberty. They mature or they start their activity, mature and start activity after puberty. These glands are found in axilla, groin, and in eyelids. They are modified, it's called Moll's glands. And in external auditory canal, they are modified, they are ceruminous glands. There, like breast is also modified apocrine gland. They are also found in breast, modified apocrine gland and nipple area. Okay. It's a modified apocrine. So next, we talked about the sites of the sites where these glands are present. Next, we'll talk about how they are regulated. Sebaceous glands are under hormonal and neural regulation. So hormones, like sex hormones like testosterone, they regulate the activity of sebaceous glands, and neural control is by sympathetic fibers. And eccrine glands, they are also controlled by hormones like antidiuretic hormone and aldosterone. And they are neurally regulated by sympathetic fibers. But usually the mediator in sympathetic system is adrenaline and noradrenaline. But in case of eccrine sweat glands, then the mediator is acetylcholine. It's not noradrenaline or adrenaline. Apocrine glands are under sympathetic regulation, sympathetic fibers, which are adrenaline. They are not cholinergic. They are adrenergic. So this is all about the glands. So we talked about the type of secretion, the function of the glands, the sites where they are present and absent, and how are they regulated. Next, we'll talk about the structure of nail. So nail is it's a chitinous structure, and nail is, there are two important things. One is nail plate, and which rests on the nail bed. Okay? This is the nail plate. And when you see the side view, you can see this nail plate rests on the nail bed. Okay, this area is called the nail bed. And this nail plate is held down to the nail bed by the nail folds. And there are 3 nail folds, that is, on either sides you see the lateral nail folds, and on proximally you see the proximal nail fold. And there is a thin extension of skin from the proximal nail fold which holds the nail. Okay, that's called cuticle. Okay, the cuticle is the extension of the proximal nail fold which holds the nail plate to the proximal nail fold. And you can see this lunular structure, the present shape and lunar structure. It's called lunula. Okay, lunula is the visible part of the nail matrix, which is seen through the nail plate. Okay, it's a visible part of the nail matrix. So, where is nail matrix situated? Nail matrix is situated. Nail matrix is situated below the proximal nail fold, and this nail matrix is where the nail is synthesized. Okay, so it's a very important part. Okay, nail matrix, and there is this visible portion of the nail matrix which is seen through the nail plate is called the lunula. This is all about the structure of nail. So there was a nail plate which rests on the nail blade and it's held down to the surface by the nail folds. And there was extension of the skin which is from the nail fold. It's called cuticle. And there was a crescent shaped structure which can be seen through the nail plate. That's called the lunula, which is the visible portion of the nail matrix. The nail matrix is the part where nail is synthesized and is situated below the proximal nail fold. Okay. So last topic is the cutaneous receptors. So in the dermis, we talked about the hair follicle sebaceous glands, the eccrine glands and apocrine glands. In addition to that, there are also cutaneous receptors. There are 4 important cutaneous receptors which is seen in the skin. One is Merkel cells. Next is Meissner's corpuscles. and Ruffini corpuscles and Pacinian corpuscles. As you can see, Merkel cells are seen in the basal layer of the epidermis. So this is the epidermis, and this is the dermis, and this is the subcutis. Epidermis. This is the epidermis, below which is the dermis, and this is the subcutis. Okay. So Merkel cells are seen in the basal layer of the epidermis, and Mason's corpuscles are seen in the dermal papilla. And we talked that dermal papilla is the upward projection of the dermis into the epidermis. So Mason's corpuscles are seen in the dermal papilla. Pacinian corpuscle and Ruffini corpuscles are seen in the deeper dermis, just above the subcutis. Okay. So now we'll talk about those receptors. So corpuscles are generally encapsulated structures. They contain the capsule. Okay, that's why it's corpuscles. They are encapsulated. Whichever contains the term corpuscles are encapsulated. Merkel cells are seen in the basal layer of epidermis. Mason's corpuscles are seen in the dermal papilla. Ruffini corpuscles are seen in the deep dermis. And the Pacinian corpuscles are also seen in the deep dermis. Okay. And regarding adaptation to sensation, Merkel cells and Ruffini corpuscles are slow adapting. Whereas Mason's corpuscles and Pacinian corpuscles are fast adapting. Okay. And what are the sensations do these receptors mediate? So Merkel cells mediate touch sensation. And Mason's corpuscles, because of fast adapting, they mediate fine touch. And Ruffini corpuscles, because of the presence in the deeper dermis and they're slow adapting, they mediate touch and pressure sensation. And Pacinian corpuscles, because of their location in deep dermis and because they're fast adapting, they sense the pressure and vibration sense. Okay. So this is about the cutaneous receptors. So there are 4 receptors: Merkel cells, Mason's corpuscles, Ruffini corpuscles, and Pacinian corpuscles. Corpuscles are generally capsulated, that's encapsulated. And we talked about the sites where they're located and whether they are fast adapting or slow adapting. And we talked about the sensation mediated. So the questions can be asked about what are the types of sensation regarding their adaptability. Or they can also ask about the location of the corpuscles. Okay. So this is all about the structure of skin. Okay. This is important to understand the structure of skin, to understand the various disorders, cutaneous disorders. Okay, so if you understand the structure or the process of keratinization, it will be easier to understand ichthyosis disorders. If you understand Dermapodal Junction and the Desmosomes, it will be easier to understand the Bullous group of disorders. Okay, and if you understand about the glands, it will be easier to understand about the acne and the hyperhidrosis and other disorders of the glands. Okay, and it is also important to understand the various layers seen in the epidermis, the structures and cells in the epidermis. Okay. So now, today we talked about the structure of skin. So it was divided into epidermis, dermis, and subcutaneous tissue. Epidermis, it is of 5 layers, and they contain 4 cells. And dermis is of two layers, papillary dermis and reticular dermis. The dermal papilla is called the papillary dermis, and the dermis below that is the reticular dermis. We saw the cells and the important fibers, collagen, elastin. We also saw about the ground substance. Next, we talked about the air shaft and air follicle. We talked about the parts, cross section, types of hair, and the phases of hair cycle. And we talked about the structure of nail, and we talked about three glands: sebaceous glands, eccrine glands, and apocrine glands. And we talked about the cutaneous receptors. So the important functions of skin are the defense barrier, because they come in contact with the external surface, so they should be the first site of resistance. Okay, they prevent the loss of water and electrolytes because of the lipoprotein envelope, because of presence of the corneocytes. They help in the temperature regulation by the secretion of sweat. They also provide insulation because of the presence of cutaneous fat, and because of the presence of receptor, they help in sensing the sensations. And because of the secretion of the sebum and the sweat, they help in lubrication. And skin also contains enzymes which helps in the synthesis of vitamin D. Okay, this is all about the basics of dermatology. So please, this is all about today's lecture, and please subscribe to Nextillo channel for more updates and more discussions on the topics regarding dermatology and other subjects. And please see the live lectures daily to get more important topics. And fine, to end with, there's a question of the day. So please send answer. Please type the answer in the YouTube chat. So the question is, laminate file is seen in which layer of the Dermapodal Junction? Is it seen in Desmosome, or Lamina Lucida, Lamina densa, or Sublamina densa? Okay, please enter the answer in the YouTube live chat. Okay. So this concludes today's topic of the basics of dermatology. So thank you for your patience listening.